Please welcome Dr. Hemmel.
So what I thought I would do is walk you through some of the key points in this paper, so when someone asks you, well, what is it about, you can intelligibly talk to them about it and really inform them about how this work has sort of advanced to this stage. So here's a little snapshot of what we've discovered in this paper and how we're putting sort of these ideas together. So the take-home message, the highlight of this paper, is that the length of meditation practice that people undertake influences their susceptibility and management to COVID-19 disease. And I'll talk about how we showed that and what we discovered to create that data set. That the blood plasma from meditators can limit viral infection in cultured lung cells, and I'll talk about why that's important. We'll talk about meditation elevating the SERPINA5, this particular factor, as a major player in coagulation and immune response. And this appears to be a molecule that's critical to COVID infection. And then the overall conclusion that we wanted to reach in this paper is that if you create and activate a regular meditative practice, that you can improve your well-being by modulating your biological process. And this is the paper that starts to unravel that, right? So this has been a story. And so when I talk to people about this, in my mind, it seems like 15 years we've been doing this. And so I had to put a timeline together. It hasn't been 15 years. And so this all started in February of 2020 in Indian Wells. It's the first time I met Joe. And this is where we did our first study. It was about 30 individuals that were selected as part of the Indian Wells study. We collected blood from them. And then something crazy happened in March and April of 2020. The world started to go through this global shutdown. As this shutdown began to drag on and we didn't know when it was going to end, people started thinking about ways to reinvent their laboratories and become relevant. So we had this blood from February. I had this guy that kept calling me, and it was like, I didn't even have his name on my phone, and it turns out it was Joe Dispenza. Like, so have you done anything with that blood from Indian Wells? I'm like, I don't think so. And so we started looking. We started doing some preliminary analysis, and because we were starting to come back in a small fashion into the lab, we had to pick someone to start with. And so 1 is a good number, right? And so we started with subject 001. Just started playing around in a variety of assays in the lab. And we had this idea that we wanted to start doing something COVID-related. One of my big collaborators on campus runs the viral vector core. And so I talked to Atsushi and said, hey, we want to work on coronavirus. I know we can't work in the BSL-3 facility because there's a line out the door to use it. But I've read about these pseudo-viruses that we could use in my lab, right? They're fake viruses that look like coronavirus on the surface, but they don't infect or do any pathogenic things to them, to cells and bodies. And so he's like, yeah, I can make one. And so we designed the first spike pseudo-virus, and he got to work. We threw subject 001's blood in a dish. We exposed it to virus, expecting cells to turn red, and I'll describe why the cells should turn red, and none of the cells turned red. And so it was a head scratcher, right? And so Juan had done this experiment. I'm like, I don't believe you, go do it again. So he does it 5, 6 times. Every time the cell doesn't turn red. Why that's important is that suggested in this assay that the cell is not being infected. There's something inside the blood of subject 001 that was preventing this virus from getting in. This then started us on this path. We started looking at all of the samples from Indian Wells. As we started coming out of the pandemic, there were 2 events, one in Orlando, one in Denver. We recruited more subjects, we had more samples, the numbers got a little bit bigger. And then we had a full story ready to go January of 2022, where we presented this as evidence as the loudest voice. And a year and some many months later, you saw the paper. So to get from start to finish in 3 years is amazing, right? And I'll show you the numbers in this paper that we have. It should have taken a lot longer, so we've gone very fast. But again, we would have liked to have gone faster.
001 was the first person that was in line for the research. So he was the first one, so he was 001. When we saw the initial blood values, his metabolites before the event and after the event, he looked really sick at the beginning of the event, and at the end of the event, his blood values changed dramatically, and he happened to have a very profound moment for himself, a few profound moments for himself. It was really changing his biology. There was information in his blood that wasn't there before those moments. So now we're going to expose his blood to a pseudo-virus and see what happens. And none of the viral tissue would enter the cell. It just wouldn't get in. And so no matter how they did it and tested it, it still showed the same results. And so then now we were kind of even more surprised at how powerful his blood was, not just the metabolites that weren't there before, but wow, there's information in there that's causing some type of a resistance or immunity or resilience to the exposure of a virus that normally enters a cell pretty rapidly.
So a little bit of evidence for each of those pieces. And so the initial highlights that I showed you talked about more of a global effect. That's not where we started, right? We had this blood from meditators, and this is where the work actually started, and it was a little backwards in terms of how it's presented in the paper. And so I want to show you in the time scale of how we did things and what was the aha moment that led us to the next step. So this first statement, blood plasma from meditators can limit viral infection in cultured lung cells. So how do we show that, right? We had access from this initial study in Indian Wells, as well as Orlando and Denver, to controls. These are individuals that were vacationing, relaxing controls that were not meditating. Access to blood from novice individuals, people who had never been to a live event. This was their first live event they were at. And so they had that skepticism and all the other things that were happening, and they changed in unique ways. And then we had access to blood from experienced individuals that had been to at least 3+ events and ones that practice a daily routine. So we could really look at the full spectrum of experience. Someone who's not meditating but relaxed, someone who's just started, and someone who does this day in, day out, right? So their blood should be very uniquely different in this setting. So then how do we test this in the system? So this is a cartoon and then an actual electron micrograph of the pseudo-virus that we created. So the way that SARS works is that there's these spike proteins on the cell surface. These spike proteins engage with lung cells, and then this is what causes infection. And so what we can do is we can engineer a fake virus to have spike from SARS-CoV-2. The inside of the virus, we then eliminate anything toxic out of, and we replace with a red reporter protein. So when this fake virus, which looks on the surface like SARS-CoV-2, and so this is the electron micrograph. So a coronavirus is called a coronavirus because it looks like a crown, right? So if you squint, this looks kind of like a crown with some points around it. And so when this pseudovirus gets put onto lung cells, we let it sit on there for 24 hours. The idea is if that virus can get inside the cell and penetrate in, that red reporter starts to express and the cell turns red. And so what you now see is in 24 hours, the entire plate starts to look like this, right? So this gives you a sense of infectivity. And so by tracking the amount of red on a plate in 24 hours, you can tell how many cells were infected. So that's the main assay. So then the question becomes, since we can't study this in a human being at the meditator level, how do we create a meditative experience in a cell, in my lab, in a dish that's never seen meditation? Tall order. So if you buy into this idea that your brain is creating information, that information is being transferred to your body, then, and I hope I convinced you yesterday that there is a lot of information in that body, and our capture of that is the blood. And so what we hypothesized in this is that information that that meditator created was captured in their blood, which we can isolate in their plasma. So then to give the cell the experience of meditation, we can expose that cell to the blood of that meditator. That information now is going to change that new life form, which is the cell. So the assay that was developed is the lung is the primary site of infection. The major cell that sees COVID first, SARS-CoV-2 first, is the lung epithelial cells. I can buy lung epithelial cells from a company. We can grow them on these culture plates. And then we basically have them meditate in the lab by exposing them to plasma from a control meditator, a novice meditator, and an experienced meditator. So they live in this environment for an hour, and then we challenge them. They're given that pseudo-virus. We let the pseudo-virus sit on there for 24 hours, and then we look at red signal. And what we saw was confirmatory of 001. In control subjects, there is no significant effect before and after. So the before is the blood sample taken before the event starts, and the red is the blood sample taken at the end of a week-long retreat. The red signal basically goes wonky in all different directions. And this was 23 individuals we tested. In the new and the experienced meditators, over 40 people in each group, there was a significant suppression of red signal in both of these individuals. So there's something floating around in the blood of these individuals that prevented that virus from going inside and creating this red signal, right? So this is the first evidence to suggest that a meditator at a week-long event is creating an environment in their blood that prevents viral entry for SARS-CoV-2. Science is unique in the sense that if you're going to show it one way, you got to show it another way. And so pictures are worth 1,000 words. This is a scanning electron image of pseudovirus on top of these three kinds of treatments. What you notice characteristically different about the control versus these is that this is less fuzzy, right? So the reason why this is less fuzzy is because the virus is somewhere else. The reason why these two are fuzzy is the virus is sitting on top of the cell. And so what you can do is look inside the cell by doing something called transmission electron microscopy. And so when we slice through the cell and look inside, what we notice is that in the control treatment, all of the viral particles are sitting inside the cell. There's less so in the novice, and there's almost nothing sitting inside the cell in an experienced meditator. It's all getting trapped on the cell surface. So whatever those factors are that these individuals are creating is preventing that virus from engaging into that cell. It's sitting on the cell surface, it just can't get in. So this is where most papers would stop, right? And this is where we would have failed. The comment back from the editors and reviewers of the paper would be, it's epiphenomenon, right? You've discovered something, but there's no mechanism. How do you know this is real? There's got to be some way that this is affected. And so we started scratching and looking a little further. So the other statement that's made in this highlight is that meditation elevates SERPINA5, a major player in coagulation and immune response. And so one of the things that we like to do in the lab is break things. And this is how you figure things out. Taking the whole and breaking it into the unique parts to then understand what's involved. And so one of the classic experiments that we did is, could we destroy the sample to see what something is not or is to figure out where we should head? And so the first experiment that any lab would do to destroy a blood sample would be to heat the sample. So this is like a denatured egg protein kind of thing, right? So when you cook an egg, all of the protein gets denatured and is no longer functional. What's inside blood is a lot of proteins. And so the idea was, if we heat that plasma in that blood and we denature all that protein, is the effect still there? So now when we put plasma before and after that's been heated on this same assay, we see that that effect's lost. So there's no longer a significant effect of creating that resistancy to that viral infectivity. What this suggests then is that the culprit in creating this effect in the meditator is a protein, right? You destroy all the protein, the effect goes away, so it's got to be a protein. So this then limits where we start to look. And so we have lots of ways to look at proteins. And so I have a collaborator that's a floor down. We sent him what's called an immunoprecipitation. We basically fished out all of the proteins that interact with this pseudovirus and this complex. He then purified this mixture, put it on his mass spec, and identified all of the peptides and proteins that came out. This was the funny network that came out. So these were all of the proteins that could be involved in creating this meditation protective effect. When we started looking at the list of 107 proteins that came out, one of them was really important, and this was SERPINA5. This is a protease inhibitor, and it's involved in a lot of different processes. And so where this works is that SARS binds these ACE2 receptors. This protein called TMPRSS2, which is a protease, chops the binding of specific proteins, and it allows the virus to internalize. What SERPINA5 does is it acts as an inhibitor of this protease, because it's a protease inhibitor. And so now the virus can bind to the surface, but it has no way of getting in because it can't complete that final step, right? And so this was the hypothesis that we had. This protease inhibitor we discovered was targeting this protease. So then how do we prove this out? So when we look at SERPINA5 levels, they're significantly elevated in experienced meditators. When we put the blood of an experienced meditator versus a control onto an assay that only has TMPRSS2 protein in it, we can get an inhibition of TMPRSS2 activity with plasma from an experienced meditator, but not from a control cell. Interestingly, when we take plasma from a vaccinated individual, there's absolutely zero effect in this assay. So they're working through a completely different mechanism. So meditation is doing something that vaccination doesn't do. Unique. So then the idea is, if this is true, that SERPIN levels are elevated, SERPIN inhibits TMPRSS2 activity, could we just buy SERPIN? And so we ordered pure SERPIN, and we put it on the same assay, and we get the same effect. When you put pure SERPIN on the same red assay, it limits the infection of that pseudo-virus, just like blood from a plasma from an experienced meditator. We're not only showing that there is this unique effect of limiting it, but we have a molecular target. Here's the protein that a meditator elevates, and it makes biological sense. What's interesting is that as we were finding this out, there's a lot of papers in the literature around COVID that had already discovered this mechanism, not in meditators, but had figured out that SERPINA5 could be a target to treat COVID-19 disease. The companies are developing an exogenous drug. We're showing you can do this by closing your eyes for 35 hours, which is pretty profound. So the final piece of all of this, and what's presented in the paper first, is we're sort of scratching our heads and sort of kicking ourselves like, is this real, right? The biology makes a lot of sense, but in practicality, is this happening in people that practice meditation? And so this final statement, the length of meditation practice may influence susceptibility and management of COVID-19 disease. So how do we do this? We designed a survey, and it asks a lot of questions about the type of meditative practice you do, how much you meditate, what your demographics are, where you're living, all kinds of things. We got back a huge response. So this is a color coding of all of the places across the world where people responded to this survey. And so by having this huge amount of data to start looking at the experience of meditators and where they had responded to our COVID survey, we could start building a world map of this relationship between meditation practice and COVID. In this survey, we had 66 countries that were represented, and multiple of these regions we could use because we had enough people to do statistical analysis. So what emerged out of this is COVID-19 rate was significantly 50% reduced in people that had an average meditation experience life cycle of more than 6 months. So the more experienced of a meditator you are, the less likely you were to get COVID-19 disease, right? So this, in practicality, proves out the biology that we were showing in a dish. People were experiencing the same effects if they were doing meditation in their entire body, not with the fake virus, but with the real virus. So we went on to show a couple other things. It turns out that we asked about frequency of meditation. And just like taking a pill, if you take it every day, if you're diagnosed with a particular disease, you have a better effect than if you forget it once in a while. And so what we noticed is that individuals that meditate every day have a much lower risk of COVID-19 than individuals that meditate once a week. So there is a dosing effect as well. Not only how long you've meditated, but how much you meditate creates this protective effect. So this pans out as well. So in these individuals that we captured blood from, it's an insane amount of meditation they're doing. It's a week-long meditative retreat. The dosing effect holds true as well in a practical population. The final question was, and people have experienced this, and I've talked to a lot of people about this, just because you meditate doesn't mean you're not susceptible to COVID. So the other big part of the analysis was, well, what happens to individuals if they meditate and get COVID? What we noticed is that a couple of the symptoms, and this is, so on each of these axes, you look at experience in the community, and 1 means that 6 months plus, and 0.5 means very little meditative experience. So brain fog is one of the major symptoms that people experience. So in individuals that meditate and that have this experienced meditative practice, there's virtually no brain fog in those individuals, even if they get COVID. In individuals that meditate, there's almost zero congestion, right? So you've been diagnosed with the disease, but you're not expressing the symptomology as highly. Loss of smell is dramatically reduced. And then what was really striking was your length of COVID mean infectivity was significantly reduced as well. So this suggests that there is this benefit of limiting infection, but also driving it to a much faster resolution with less symptomology if you're a meditator, which was quite profound. So taking all of that together, I think we can clearly say that a regular meditation practice can improve the well-being by modulation of biological processes. And this is definitely true for COVID. I think the message of this paper is going to be read in a couple different ways. I think people are going to focus on that COVID aspect. And I think where I would like them to focus is really on the meditative aspect and what it's doing to biology. This is the first layer of that onion, and it just happened to be COVID because it was opportune, and this is where we're headed. If we expand our mind and think about this, right? The meditator is not just elevating SERPINA5, there's thousands of other things that are there. And where we think this really is leading to is, I think the blood of meditators has the cure for cancer, neurodegeneration, autoimmunity, anxiety, depression, PTSD, you name it, it's there. And so that's where we're headed, is we're approaching it the similar way, creating cell models to test this out, and really figure out a way to attack this from a mechanistic perspective. And I think we're going to uncover a lot.
Thank you so much.
Yeah.
Did you know that all of us share a power within? Meditation can help us heal and thrive, but proving this is a challenging task. Groundbreaking research is showing ordinary people doing the extraordinary, changing their physiology, changing their biology, and changing their lives, not with a pill, but with the power of meditation. Evidence is the loudest voice, but rigorous scientific research can be challenging. Advancements in science and technology often require significant investments of time, resources, and money. Thanks to Dr. Joe and Inner Science Research Fund, we're able to conduct large-scale studies that were inconceivable a few years ago. The research that Dr. Joe funds at his events has generated tens of thousands of biological and physiological samples. That's billions of data points that need to be analyzed by Dr. Himmel and his team at UC San Diego. Inner Science and its donors make the analysis possible, which drives additional research publications, which spreads the word about the powerful potential within each of us.